A very good evening and welcome to one and all present here for this webinar series DENTOPHARM 2022 on targeting epithelial to mesenchymal transition by small molecule inhibitor Fisetin in lung cancer. I am Dr. Ramya, part of Dental Education Unit and Senior Lecturer at Sri Balaji Dental College and Hospital. We'll be highlighting you about this program today. I would like to welcome our chairperson, Dr. Srinisha ma'am, Registrar Dr. Bhuminathan sir, Principal Dr. Jimson sir, and Dr. Sumati Jones ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati ma'am to share a few words on today's webinar.
Good evening to the delegates present over here. As you all know, our dental webinar series, DentoPharm, is all about the uh, research in diversified field. Today's topic will be on lung cancer. Dr. Sabha Tabasam has accepted our invitation and will be delivering lecture on lung cancer uh, and the drugs, mainly Fisetin. We all know about Fisetin, its antioxidant property, its anti-inflammatory effect, senolytic property, and all those. She'll be explaining in detail about the particular drug in lung cancer, its mechanism. Over to Dr. Sabha Tabasam.
Good evening, everyone. I would like to thank Dr. Jimson, Dr. Jones, and all the of the webinar series from Balaji Dental College for giving me the opportunity to speak to you. Before I start, I would like to briefly mention I'm a research fellow at Dana-Farber Cancer Institute under Harvard Medical School in Boston, and it's always a pleasure to talk to you. So on with my presentation here. So as mentioned by Dr. Jones, the topic of my talk will be targeting epithelial to mesenchymal transition by small molecules, that is Fisetin, which is actually for implications for cancer invasion in metastasis. The cancer model that I have worked closely with is lung cancer. Lung cancer is a leading cause of cancer deaths worldwide. Every year, lung cancer causes around 1.6 million deaths, which is more than breast, colon, and prostate cancers put together. Lung cancer accounts for about 7% of all new cases. And the regions where it's most or highly reported is Mizoram in both the sexes, males and females. The main cause of lung cancer is active smoking, followed by radon gas in homes, workplace exposure, smoking, and treatments and genetics. It is estimated by the Global Cancer Report that by 2030, the number of new cases being diagnosed for cancer will go up by 50%, and the number of deaths due to cancer will go up by 60%, accounting India at 7.2 and 8.3 in terms of increase in mortality. The 5-year survival rate, which is a measure by which we try and establish that what is the progression of cancer? Generally, for every cancer, there is a 5-year survival rate, which tells us that if somebody is diagnosed with a particular type of cancer, in 5 years, what is the expectation? In terms of lung cancer, if the diagnosis is made at an early stage, which is only about 15% of being diagnosed at early stages, the 5-year survival rate is 54%. However, majority of the cases are diagnosed at a late stage, that is about 57%, because of which the 5-year survival rate drops to an alarming number of only 4%. The challenge is here that the majority of lung cancer patients have late-stage diseases, and that is not curable by current therapies and responsible for the low survival. A large percentage of those diagnosed with early-stage diseases eventually, if not cured, experience recurrence of diseases and land stage. The treatment of advanced lung cancers is improving, but standard treatments such as chemotherapy and radiotherapy have limitations. And there are multiple and different strategies required to improve this 5-year survival rate. When we talk about cancer, we are generally talking about the spread of cancer throughout the body, which is known as metastasis. It is one of the major cause of treatment failure in cancer patients and results in cancer-related deaths. Metastatic tumors are not only difficult to treat with conventional surgery or radiotherapy due to their anatomical diffuse localization in different organs, but in most cases are found to be resistant to the therapeutic options or the chemotherapeutic regimens available right now. The molecular mechanisms underlying cancer metastasis and chemoresistance are mostly unknown and are the bleeding edge of cancer research. There are more than 200 anti-cancer drugs that have been approved, but none of them specifically targets cancer metastasis. Metastasis is a complex phenomenon which takes place by multiple pathways, and one of the pathways that have spotlight from quite some time is known as epithelial to mesenchymal transition, which is has been recognized or acknowledged as an important event to strengthen the metastatic availability of the cancer cells. Epithelial transition. I would like to define it for you that it is a biological process that allow epithelial cells, which are supposed to be which are supposed to constitute the tissues and have a very well-defined structure. They are known as polarized because they have a edge, they have a regular edge, and they are found in sheets and found to be they form structures, very well defined, very well organized structures. However, if they undergo this transition, that is from epithelial to mesenchymal transition, they shed properties and become more motile, and they have access to invasive migratory capacities. And these cells and the mesenchymal state of cells are found to be resistant to apoptosis, and it increases the production of the extracellular matrix proteins. This phenomena which is exclusive in cancer. This is something we see in the fetal stages where the organs are being formed, where there's a major migratory events of cells to constitute the different layers of the mesoderm. And apart from that, in the physiological scenario, uh, the repair process that takes place every time they are wounded involves epithelial to mesenchymal transition. So epithelial to mesenchymal transition is a physiological phenomenon which takes place in the body even without any disease. And it is important for healing. But cancer cells gain access to this particular pathway and use them to spread throughout the body and establish secondary tumors or tertiary tumors. Tumor prevention is defined as a use of natural synthetic chemical agents to reverse, suppress, or prevent carcinogenic progression to invasive cancer. So chemopreventive agents can be used in throughout the stages of cancer, starting from initiation to promotion to progression. There are various class of these agents depending upon their property. Be categorized into antioxidants, antimutagens, antiproliferative, anti-inflammatory. The chemopreventive agent that I have worked on is Fisetin, as introduced. It is a naturally occurring flavonoid which is found in fruits and vegetables. So the most common sources includes onion and strawberries. Dietary Fisetin possesses antioxidative, anti-inflammatory, and antiproliferative activities in a variety of cancer cells, and it has been well reported. Although Fisetin is proposed to inhibit the growth of various cells or cancer cells by apoptosis, the precise impact and related molecular mechanism of Fisetin on migration and invasion in context of the epithelial to mesenchymal transition is unexplored. So here, EMT takes place at the very initial stage in which a primary tumor under the effect of multiple growth factors and cytokines take this mesenchymal state, which has a metastatic potential and which is capable of spreading throughout the body. And once it reaches another site, it undergoes reverse of EMT known as mesenchymal to epithelial transition to establish a distant metastatic colony. So there are multiple stages on which we can target the EMT pathway. The first one is when the primary tumor itself is undergoing, and it is in the very initial phase. The second one, when the advanced stage is being established and the cancer cells are ready to spread throughout the body. Though I have explored this separately with the best interest of today's time, I'm seeing only the targeting at the advanced stage. To note, as these cells undergo EMT, there's an increase in drug resistance, which makes them very difficult to target by the conventional treatment options available. Given the role of EMT in promoting chemo-resistance, invasion, and specific targeting of EMT could potentially serve to decrease metastasis and overcoming drug resistance. As mentioned, we explored the effect of Fisetin on a mesenchymal cell line here. We chose H1299, cell lung carcinoma cell line. It is an advanced representation of the tumor. It is negative for E-cadherin. So E-cadherin, also known as epithelial cadherin, is actually it's a junction protein. So when cells establish forms different structures, there are various proteins by which they interact with each other, which support them structurally, functionally. So one of the cadherins are basically proteins by which cells bind to each other, and them it's like interlocking. So it helps them form that sheet or the layers on the basement membrane to establish a particular morphology. However, when epithelial to mesenchymal transition takes place, the epithelial properties are shed, and one of the hallmark is downregulation of cadherin. As mentioned, mesenchymal cancer cells have the ability to invade, to migrate. They are resistant to chemo and radiotherapy. They are multipotent. They are capable of differentiating into different cell lineages. Ability to self-renew. They can make multiple copies of themselves. They are capable of evading the immune system, and they show anoikis resistance. So just to explain what is anoikis here, the epithelial cells are supposed to be alive when they form or they found an attachment surface, which in physiological context is a basement membrane. Every time they detach from basement membrane, there's a apoptotic switch that is turned on and they die. However, in cancer cells, when they undergo T, they gain access to anoikis. This is something only mesenchymal cells which are present in physiological scenarios. Every time we are wounded, the skin needs to come back and join together to close the wound. There is an EMT that is triggered which allow the healing process place. In that case, the cells which do not attach to basement membrane gain the ability to survive until and unless a new basement membrane is being established so that the repair is completed. However, cancer cells take advantage of this particular process, gaining access to EMT, and they survive even without a basement membrane. The reason why they can travel all the way in the bloodstream or in the circulatory system and gain access to different sites. So to begin, I would like to break it down, the study. Like when we have such a problem, we try and define a strategy or a pipeline around these kind of studies. So we start, since we are working with a chemopreventive agent, we start working by standardizing dosage. So we started by screening for non-cytotoxic concentration of Fisetin, because the reason that we are emphasizing non-cytotoxic, because we are not trying to kill the cells here. They are already very potent and very anti-tumor drugs available in the clinic, which are capable of killing the cancer cells. But at the same time, cancer cells which have undergone these advanced signaling pathways or have gained access to this fetal pathways are not capable of being targeted by the cytotoxic agents. So here we are trying to propose that if Fisetin is capable of inhibiting EMT and preventing the metastasis, maybe it can be used in combination with the mainstream drugs which are capable of cells. So we broaden our targeting abilities in terms of we have a drug which is capable of killing cancer cells, and at the same time there is a drug which will prevent the spread, and this will further potentiate ideally the effect of the first drug. So we start at the non-cytotoxic concentration of Fisetin. We chose a range of 0 to 50 micromolar, and we found that doses beyond 10 micromolar were capable of inducing cell death at both the time points 24 and 48 hours. So we took note of 10 micromolar doses, and we started exploring the anti-EMT activities of these particular doses. We performed a wound healing assay, which is generally used to study the migratory abilities of the cells and generally to which are capable of inhibiting the migration of these cells. And we screened the doses 5 and 10 micromolar, and out of the two doses, we found that 10 micromolar was most effective. Here you can see, in spite of at later time points from 12 to 10 micromolar, significantly inhibited the migration or the healing of this particular scratch. Next, we went on to explore the anti-invasive ability for this. We performed a point-in-chamber assay in which we use an matrix known as matrigel, and we plate our cells over the matrigel and treat them with it. And we put a chemotactic agent at the bottom of the valve to allow the cells to get attracted. So when all these dynamic movements are taking place in a tumor microenvironment, there are a lot of cytokine gradients being established. The reason why a cell gains a direction for its movement, we try and recapitulate these conditions in labs and establish such simple cell-based assay. And in our quest to establish, sometimes we oversimplify things because it's difficult to create a tumor microenvironment. There are multiple cytokines, multiple cell types being involved. So we do study them in isolation, but this is how we start dissecting an agent's property. We found that 10 micromolar Fisetin was capable of inhibiting the invasion of these cells, and there were less cells invading when treatment was performed with 10 micromolar Fisetin. Next, we started studying the phenotypic markers for EMT. So, as mentioned, in EMT, cells have epithelial properties and gain access to mesenchymal properties or mesenchymal markers. In this particular cell line where EMT is already established, because this is an advanced cell line, there were very less epithelial marker. And we found another junction protein, and this occluded. This is also involved in gluing cells together, if that is something you can relate to. So this helps in establishing a sheet-like structure so that the cells are non-mobile. And such glue allow the cells to separate from each other, and gaining properties like annoyance resistance allow them to survive in these conditions, the reason why they gain access to circulation eventually and landed forming a secondary tumor. So here we found that, as you can see, the expression of this was very low in the control cells. However, treating with Fisetin showed an increase in the expression of zonoclidin, suggesting a subtle shift towards an epithelial phenotype. However, in terms of mesenchymal, we look for vimentin, which is an A3 filament, which allow the cells to be more flexible so that they can move through different spaces. Here, the control cells were expressing a good amount of vimentin. As you can see, there's a signal. However, 10 micromolar Fisetin decreased the signal, which was a decrease in the expression of vimentin. So the mesenchymal counterpart of E-cadherin is known as N-cadherin, which comes from neuronal cadherin. It is also a cadherin which allows the cells to attach to each other, but N-cadherin is more, it's more of a more labile interaction. That is, it holds the cell together but does not let them to form structures, a very well-defined structure. It's like when cells are bound by N-cadherin, they can move, but they will move as a group. They are loosely attached to each other. So N-cadherin is another mesenchymal marker which went down with Fisetin treatment, suggesting that their Fisetin was capable of inducing a subtle epithelial stage and decrease the mesenchymal H1299 cells. We further checked for this expression by immunoblot assay, and by immunoblotting we found that 10 micromolar Fisetin was more effective compared to 5, and there was a decrease in Fisetin. There was a decrease in beta-catenin levels. From that, when cells are moving, so in tissues also, when these cells are organized in such a well-defined structure, there are a lot of ECM that surrounds them. When these cells gain access to a mobile or a motile phenotype, start secreting a lot of proteins which are capable of digesting the ECM and allowing them to move through it. So the most popular zymogens, known as zymogens, which are known in lung cancers, are MMP9 and MMP2, and they go and cancer cells gain access to a motile phenotype. However, Fisetin treatment, especially again, 10 micromolar was most effective out of the two doses selected, decreased the expression of MMP9 and MMP2, suggesting that the EMT is being targeted by Fisetin. At the same time, it is the range, the downstream signaling is being also affected. To check whether the Fisetin treatment was able to induce of E-cadherin, we performed the ICC assay, and we found that there was a very subtle increase in E-cadherin expression, which was mostly in the perinuclear space. And the immunoblot analysis of this did not really which suggests that E-cadherin somehow was not induced. So in advanced stages, when EMT is already established, there are a lot of epigenetic signals that are being modified. So this suggests that Fisetin is working at an epigenetic level when it comes to E-cadherin. We also check for another marker, SMAD4, which comes downstream to TGF-beta. TGF-beta is one of the most popular inducer of EMT in tumor microenvironments, and there are multiple trials going on. Fisetin treatment shows a decrease in SMAD4 activity, which comes downstream to TGF-beta signaling, which may explain that why we were seeing a decrease in mesenchymal phenotype. Another marker, beta-catenin, which is known to be even drug resistance, was downregulated by Fisetin treatment. And in line with beta-catenin, NF-kappa B is also known to involve in chemoresistance. And the decrease in NF-kappa B, especially in the nuclear compartment, was observed with Fisetin treatment, where it works because it acts like a transcription factor. So later on, we checked multiple signaling pathways which are known to be involved in EMT. The interesting thing about EMT is it is downstream to multiple signaling pathways. So if you have one signaling pathway, the other signaling pathways are available to gain access to EMT, and this is known as EMT module. So we need, there is a need of agents which targets multiple signaling pathways to avoid redundancy. Challenges that is being faced in clinics is when people study one drug, one target, that there are redundant pathways or redundant targets. If you block one, it is replaced by the other pathways. Giving so many drugs at the same time risk the toxicity issues that we generally hear and try and work around in chemotherapy. Thus, having agents which targets multiple pathways working redundantly is something very desirable. So we are studying the NF-kappa B and STAT3 pathways, which are known to be involved in resistance, and we found that there was a decrease in their overall levels in the whole cell lysates. Later on, we further dissected their expression in cytosolic and nuclear compartments, and we found that in the nuclear where they actually work and transmit the signal, there was an overall decrease in their levels with Fisetin treatments. In terms of beta-catenin, depending upon its location, is categorized as epithelial or when present in the nucleus, it is a mesenchymal marker, and when present at the periphery of the cells, that is the plasma membrane, it is known as an epithelial marker. So, very briefly, it is present at the cytoplasmic domain of E-cadherin. Whenever there is a loss of E-cadherin, that is the EMT process takes place, beta-catenin is released and it enters the nucleus where it mediates the expression of multiple genes which are capable of inducing or providing a mesenchymal phenotype to. So here we found that beta-catenin pathway was also being targeted and there was a decrease in the nuclear compartment. We also check for total EGFR, which is known to mediate chemoresistance, specifically in the nuclear compartment. It is an RTK. It is interesting that RTKs in cancer cells are found to be localized in the nucleus because these are receptor tyrosine kinase. They are supposed to be present at the periphery. And one of the reasons, because there is a class of chemotherapy known as RTK inhibitors, which are actively used in the clinic. And eventually, there's a lot of research still ongoing around resistance. And one of the pathways that were found that these RTKs somehow are present in the nucleus instead of the periphery. The inhibitors do not have access to them because they are mostly present in the cytoplasmic or the extranuclear compartment and are not able to inhibit, accounting for the resistance. So we checked for total EGFR because it was an advanced cancer cell line. We were expecting that there would be some, and we found that there was some nuclear level. There's a subtle decrease in the total EGFR levels in the nuclear compartment, suggesting again the reversal of the mesenchymal phenotype with Fisetin treatment. Since we saw the decrease in the nuclear compartment, we were interested in seeing what's going on at the periphery of the plasma membrane, and an increase in the beta-catenin level at the plasma membrane was observed, further explaining that there is a shift towards more epithelial phenotype here. Next, we studied whether Fisetin was of providing functional consequences to all these molecular mechanisms that we explored. And with us, we did a soft agar assay, which mainly studies the anoikis resistance, the anchorage independent cell death. Here we found Fisetin treatment decreased the number of colonies that H1299 was capable of forming on the soft agar, suggesting that there was a decrease in the stemness and there was a decrease in the ability to form these colonies with that. Apart from that, we found that the size of the colonies were reduced. The cells were not capable of forming such large colonies as you see in the control when treated with Fisetin. With all these being said and done, we later on to test a combination of Fisetin and erlotinib. Erlotinib it's an FDA-approved drug which is being used to target EGFR signaling, and we wanted to check that the decrease in mesenchymal phenotype resulted in any increase in activity, and we found that doses as low as E5, as you can see, erlotinib 5 and 10 micromolar. 10 micromolar was more cytotoxic. However, we did observe some cytotoxic with Fisetin 10 also, but the combination of two made even the 5 micromolar dose, which was not cytotoxic, it made it cytotoxic when given in combination with Fisetin, showing an increase in the sensitivity of the cells. So to summarize, Fisetin inhibits the migration and invasion of H1299 human non-small cell lines at non-cytotoxic concentrations, that was 5 and 10 micromolar. It targets multiple signaling proteins, including NF-kappa B, beta-catenin, nuclear EGFR, STAT3, known to mediate EMNS and drug resistance in H1299 cells. Fisetin decreased the anoikis and chemo-resistant in H1299 cells. All this resulted in increase in the sensitivity of these cells towards the anti-tumorigenic effects of even mainstream drugs like erlotinib. This is a summary of my findings. Thank you, everyone, for listening. I'm ready to take questions if you have any. Thank you, everyone.
Thank you, ma'am. Thank you, ma'am, for conveying your knowledge to this presentation. I would wish to give an opportunity to the participants to know about the speaker a little better on her achievements. Dr. Sabha Tabassum is a postdoctoral research fellow at Dana-Farber Cancer Institute, Harvard Medical School, Boston, in the United States. She has 8 peer-reviewed articles published in various publications, including Nature Communication Journal and Journal of Clinical Investigations, with a high impact factor 19.4 and 14.9. And she also has US patent filed on her research work, vasculogenic fibroblasts. She was awarded first McNaught Fellow in McNaught Fellowship 2022 at Dana-Farber Cancer Institute, Boston. Earlier, she was awarded A.R. Rao Young Research Award for oral presentation at International Symposium on Cancer Prevention and Treatment in 2018. and also received Sakura Scholar Award in Japan Asia Youth Exchange Program conducted by Kohima University, Japan. She obtained her Master of Biochemistry from Hamdard University, New Delhi, India, and also bagged University Gold Medal and doctoral degree on targeting epithelial mesenchymal transition by small molecules implications for preventing cancer invasion and metastasis from Jawaharlal Nehru University, New Delhi, India. Later, she moved to United States to pursue her postdoctoral research fellowship at Ohio State University, Columbus, and also in Indiana University. Her technical expertise are on cell culture, protein analysis, gene analysis, immunology, cytotoxicity analysis, microscopy, animal handling. She is also member of American Association of Cancer Research, the Society for Melanoma Research, and Society for Immunotherapy of Cancer. Ma'am, can we go to the question session, ma'am? Over to Sumati, ma'am, for the question session.
Okay. Okay. I hope you can hear me. Okay. Thank you, Saba. Thank you so much for enlightening our students. We have few questions in our question box. Okay. Can Fisetin be taken as prophylactic drug?
I think um, there have been some reports of prophylactic uses of Fisetin. I have not explored its prophylactic activities, but yes, I am aware that there have been mentions and research on the prophylactic uses of Fisetin.
Good. Is there any drug interaction with Fisetin?
Yes, there have been reports of drug interactions with Fisetin, but see, when we are studying, just to mention something that I should have mentioned in the earlier stages also, when we are studying chemopreventive agents, the information about chemopreventive agents or especially the traditional medicine is that somehow they have a differential approach, which is not seen in the chemotherapeutic drugs, that they are capable of distinguishing normal cells and cancer cells. So every time these agents, we firstly screen them on normal cells and we establish their cytotoxic or toxicity profiles. And when we actually establish different doses, we are always supposed to screen the same doses on normal cells also. If there are drug interactions, the drug interactions that we study are towards the enhancement or synergistic effects. We are not saying that there has to be an interaction to negate each other's effect. But yeah, there are drug interactions with Fisetin.
Thank you. Measures to increase the bioavailability of Fisetin.
That's a very good question because that's another problem that we encounter when we're working with all these phytochemical agents. As I mentioned, all these phytochemicals are present in our diet, but they are not enough to bring a therapeutic effect. That's why the purified compounds are being available commercially, on which people like me are working. But at the same time, we found that their bioavailability is a big issue, which is something we encountered in some of the famous trials involving curcumin. And there have been various approaches in which people have tried to make nanoformulations, people have tried to make different delivery systems in exploring Fisetin. But yes, there have been many studies. Nanoformulations have been very effective in increasing the viability of Fisetin, and it is an ongoing battle when it comes to especially the chemopreventive agents. Bioavailability is a big issue.
Thank you. What is the role of Fisetin in aging?
So Fisetin is a very popular in terms of anti-aging drugs. If you go online, out of all the supplements that are available for commercial use, Fisetin is there. It's sitting on an Amazon shelf. It's sitting on your supplement everywhere you go because it has antioxidant properties. So yes, there have been studies in which Fisetin have been explored in the anti-aging effects.
Okay. Is it effective in other types of cancer? Sorry.
Yes. Yes, and I think that's one of the criteria. So when you are trying to study an agent, it is only successful if it actually targets a broad spectrum of cancer models and at the same time targets the pathways with multiple cancer types. Because it takes a lot of time, effort, money to bring an agent from the clinic, from the lab to the clinic, and finally to the shelf so that it's available for patient use. So if you spend so much time and energy and money, you will not put the bet on an agent which is capable of working only in one cancer type. So generally, the agents that we see, they are or that we use in the clinic right now are capable of targeting multiple cancers. But again, cancer and cancer is a very broad term. It's a heterogeneous disease. Even if there are two people having lung cancer, they might be given different drugs depending upon what is the stage of cancer and what are the driving mutations. So especially right now, we have various onco panels available. Every time a patient is attended by a doctor, they take a biopsy sample and they try and study that what are the major driving mutations, what are the major driving targets. And around that, they try to design a chemotherapy regimen in which there are multiple drug types given in combination. And that varies from person to person. So there are regimens which are approved for particular cancer types, but again, all these agents are used in multiple cancer types.
What will be the drug usually combined with this Fisetin?
Can you repeat that one more time?
What will be the anti-cancer drug commonly combined with this Fisetin?
Oh, let me make it very clear. Fisetin is still in its preclinical stages. It is being not used.
Used.
So,
Yeah.
yeah,
If at
so
all, any specific drug combinations with Fisetin you have studied?
So I have studied with erlotinib that I showed, and I have the group that I come from extensively work on this particular chemopreventive agent, and people have tried to study different chemotherapeutic drugs in combination with this. But in context of my research, because I was exploring its anti-metastatic activities, I would prefer an anti-tumorigenic, an operative drug to be combined with the dose range that I have worked with to give a synergistic effect.
Or would the toxicity of Fisetin, will it be severe or moderate or non-toxic?
So I think this is exactly what I said in the beginning. The fascination with chemopreventive agents coming from dietary sources has been always about that the toxicity levels are very manageable, especially at the dose range on which we see their anti-cancerous agents. So if I remember, the last time I checked, it was 200 mg, which is very high and which is never given in the clinic. And even at 10 micromolar, we are seeing that it is active. So it's pretty tolerable in terms of toxicity.
Good. Thank you. Thank you for answering all these questions patiently. We'll move on to Ramya again. Ramya.
Thank you, doctor. Thank you. Thank you for answering all the questions. And definitely participants could have gained more knowledge on Fisetin and epithelial to mesenchymal transition. Thank you for your time, ma'am. I would also
Thank you
like
very
to
much.
thank the institution Sri Balaji Dental College. Thank you, ma'am. I would also like to thank the institution Sri Balaji Dental College and Hospital, Dr. Sumati Jones, Head of the Department of Pharmacology, for conducting series of webinar on DENTOPHARM 2022. Principal Sir, Dr. Jimson, and the organizing committee, Dr. Sumati Jones, Dr. Satish, and Mr. N.P. Murli. A special thanks to Dr. Nishant Sheikh and Angelin from Doc Mode organization for the technical support.
Thank you once again, Dr. Sabha. Thank you.
Thank you, everyone. Thank you very
Looking
much,
forward
Ramya.
Thank you
to
for the
collaborate with you.
participation.
Same here. Would love to come back. Thank you very much.
Thank you. Thank you so much.